So let me go over the eye movement problems again. If you have a lesion in the PPRF on the left side, you can't get your eyes to go to the left. They might deviate a little bit to the right. They probably will. If you have a lesion of this left lateral rectus, you can't get your left eye past the midline to the left. It'll be deviated immediately, and to get rid of the double vision, you rotate your head to the left. With a lesion of the abducens nucleus now, we're going to have a weakness of the left lateral rectus, and we're going to have a loss of drive to the right medial rectus. The way to correct this would be to rotate your head to the left. Lesion of the MLF here, you think about this right medial rectus that's deviated a little bit laterally. The best way to get rid of it would be to rotate your head to the left. The last thing I want to talk about is the corticobulbar input to motor 7. Now remember, if we have a large lesion of motor 7, all the muscles of facial expression are weak on the ipsilateral side. That is, above the eyes, below the eyes, they're all weak. Now, the corticobulbar input to motor 7 is kind of unique. The lower part of the nucleus, say if we just drew a line like here, the lower part of the nucleus, the cells in there innervate the lower part of the face, about the eyes down. The upper part of the nucleus innervates the muscles of the forehead, I would say above the eyes. So upper part, I also call this the dorsal part. Upper part, upper face, lower part or ventral, lower face. Turns out that the corticobulbar input to motor 7 is kind of unique. The upper part of the nucleus, gets a bilateral input. So it's like nucleus ambiguous. The lower part of the nucleus gets only crossed input, so it's like the hypoglossal. So there are corticobulbars down here that get off and go up into the motor 7. Now don't forget, the upper part of the nucleus gets bilateral input, the lower part of the nucleus gets crossed input. So if you have a big cortical lesion, say on the right-hand side, here's what would happen. You would have a left hemiplegia, your tongue would go to the left, and the left side of your face would be weak below the eyes. So let's do this again. The upper part of the nucleus gets bilateral, and it goes to the upper part of the face. The lower part of the nucleus gets only crossed and goes to the lower part of the face. So if you lose your corticobulbars, if you lose your corticospinals up there, and a big lesion in the right cortex, you're going to have a left hemiplegia, no atrophy. Tongue is going to go to the left with no atrophy, and your face is going to be weak below the eyes on the left with no atrophy. Now, realize that the corticobulbars in here, if we had a lesion here, the corticobulbars to motor 7 have already gotten out. They've already done their job. So if we interrupted the corticobulbar fibers in here, we're just going to have to worry about those corticobulbars going to ambiguous and hypoglossal, and really we only got to worry about the ones to hypoglossal. You do not have to worry about the corticobulbars going up in here to motor 7 because they've already gotten off slightly rostral. But at the levels rostral to this motor 7, we're always going to have to worry about those corticobulbars. Well, now if we turn to level 6, we can see some small changes. Level 5, I always think about it as cranial nerve 7 and 6. And this level 6 here, I always think about as the trigeminal level for me. Mainly because you can see the big nerve coming in here, penetrating the middle cerebellar peduncle. So this is cranial nerve 5 here. Realize there are fibers going out here for muscles of mastication, there are sensory fibers coming in. So the ganglion would be out here. Let's take what we already know. If we come down here into this again now, this is the basilar pons, the base or basilar pons down here. Here's your middle cerebellar peduncle. This area up in here would be the tegmentum of the pons. Here's your fourth ventricle right here. So we got a lot of cranial nerve things up in here. Down here we got corticospinals, corticobulbars, and corticopontines. Now you know about the corticopontines. They go into the pontine gray, pontocerebellars. We know that. If you had a lesion of the pontine gray in here, a little virus, you'd have a contralateral incoordination. Down here, those cortical spinals, you know you're going to have a contralateral hemiplegia, contralateral Babinski. Now the cortical bulbars, they're always kind of a problem. Well, what cortical bulbars would you be cutting here? Well, you'd be cutting cortical bulbars to any cranial nerves that are caudal to here that we have to worry about. Now what's caudal to here, of course, is going to be the hypoglossal, okay, nucleus ambiguus, and motor 7. So let's do the cortical spinals. You'd have a contralateral hemiplegia. Let's do the hypoglossals. Your tongue would go contralaterally. Your uvula would hang straight. And when we get to the muscles of facial expression, we're going to have a problem with those muscles on the contralateral side below the eyes. So let's do it again. If you had a lesion in the right motor cortex, you'd have a left hemiplegia. Your tongue would go to the left. Your uvula would be straight. And you have weakness on the left side of the face, but below the eyes. There's the old saying, you can always wrinkle your forehead with a cortical lesion. Cortical spinals, cortical bulbars, cortical pontines, pontine gray. We understand all that. Now, what's chucked up into here, it gets kind of squelched together. Here we'd have some of the lateral lemniscus out here, laterally. So we know subtle auditory deficits. If somebody asked you where these axons came from, you could say cochlear nuclei or superior olive. If they ask you where they're going, you could say the inferior colliculus, which is rostral to this. Now you move out into here and look at this. Everything is kind of squelched together. It looks like a smaller pathway, but they're just they're all still in here. You have your TTT, your ALS, your medial lemniscus, and your descending autonomics. All right, so everything's going to be contra in here. That is as far as the loss, except for that pupil. It's going to be on the ipsy side. Here's your old friend, the MLF, right here and here. Medial longitudinal fasciculus. Well, if I was to ask you where these cells are going, you'd say, oh, all the way up to the oculomotor complex, medial rectus neurons. If I ask you that's where they're going, where they're coming from, you could say the opposite abducens nucleus, the contralateral abducens nucleus. If I ask you what happens if you cut it, if you cut it, well, you can't get the ipsilateral eye to go medially upon attempted horizontal gaze this way. Okay, so remember, this is going up to the medial rectus. Medial rectus is going to make your eye go this way. So when you try to look to the left, you're not going to be able to get that eye to go medially towards the left. You would not have any atrophy. You would not have any atrophy of these medial rectus neurons up here because the lower motor neurons are still alive. Also, you could get this eye. You could get this eye up here, this eye on this side. Okay, in other words, here's your MLF going up to this medial rectus neurons. These medial rectus neurons are alive and will contract, especially if you converge. Convergence, both eyes go medially at the same time. So that shows that those neurons are alive. They're getting their information from what we call the convergence center, okay, which would be not way rostral to the midbrain. So that information about convergence would come down into these medial rectus neurons and make them go medial. This pathway is for horizontal, voluntary horizontal eye movements, okay? So this pathway is cut. So when you want to voluntarily look this way with this right eye, it won't go. But if the convergence center turns on, it'll come down in here and tell those medial rectus neurons to fire, and they will. The key identifier, though, is the trigeminal nerve coming in here. So let's just take some of these sensory fibers. We already know the pain and temperature fibers, right? So they're coming in here at the level of the pons here. This is rostral pons to me. So the ganglion, the trigeminal ganglion would be way out here. Here comes that pain and temperature fiber that we picked up way down at level 1, way long time ago. There's that pain and temperature fiber, comes in, penetrates the middle cerebellar peduncle, gets in here and looks around and says, oh my gosh, I don't see any place to synapse. And then it just goes all the way down here, whoa, down into here to synapse, pain and temperature. Then, once it's in here, jumps in the nucleus, then it crosses and goes up the famous TTT. So it's going to be in here now, okay? TTT would be here. So, if you're a pain and temperature fiber, there you come in and you look around, and there's nothing there, so you go caudally. Now, if you're a two-point discrimination vibration conscious proprioception fiber, again, you're going to have your cell body out here in the Gisserian or trigeminal ganglion. You're going to come in here. You're going to look around. Darn, there's a nucleus waiting for you right here. Chief sensory 5. On this side, chief sensory 5. All right? So here's a two-point coming in. Comes in. Oh, there's a nucleus. I'm going to synapse it in it. So it jumps in the chief sensory. Now, the chief sensory nucleus has cells that are receiving this two-point discrimination, etc., from the face. And these cells are then going to send their axons across the midline and eventually going to join the TTT, but not at this level. So in other words, they come in, they synapse, but then they filter across, and then at the next level, right in here, they're in the TTT. But at this level, they are not in the TTT. So if you had a lesion down in here, okay, where you have your ALS, your TTT, your medial meniscus, your TTT is just going to give you contralateral loss, pain, and temperature. You're not going to have any two-point loss. The way you get your two-point loss is to have a lesion in the nerve, couldn't get in, or in the chief sensory. So if I had a lesion up here in the chief sensory 5, I'd have a loss at two-point, etc., from the ipsilateral side of the face. If I had a lesion here, I'd have a loss of pain and temperature, and two-point, etc., from the ipsilateral side of the face. So here's your chief sensory, two-point from the face. It's like the dorsal column system for the body. Now, here's the chief sensory, and medial to it right here is motor nucleus 5. Okay? So you got a motor nucleus 5, you got a motor nucleus 7, you got a nucleus ambiguous. And all these nuclei are innervating muscles of branchiomeric origin. So in the case of the motor nucleus 5, it's going to innervate, it's going to be lower motor neurons, whose axons go out the nerve, like this, and go out to the muscles of mastication. So, if you had a lesion in this nucleus, you have a lower motor neuron problem. You have atrophy of the muscles of mastication. And when you open your jaw, that jaw is going to deviate towards the weak side, just like the case of the hypoglossal. So, here's motor 5. There's a cell in here. Its axon goes down and out, goes out to muscles of mastication. If you have a nerve lesion or a nucleus lesion, your jaw is going to deviate towards the side of the lesion. Now, cortical bulbar input to motor 5 is good for you med 1s, all right? Bilateral, okay? So you're not going to have to worry about the cortical bulbar input to motor 5, all right? Anyway, if you're down in here, the only cortical bulbars you got in here are going to motor 7, ambiguous, and hypoglossal. Of those you got to worry about, well, most of the hypoglossal are crossed, and those to the bottom part of motor 7 are crossed. So you interrupt this, again, I kind of hate to keep harping on it, but if you interrupted these right here, you'd have a contralateral hemiplegia. Your tongue would go contra, and your face would be weak on the contra side below the eyes. You could always wrinkle your forehead. The cortical bulbars in here, the cortical bulbars have already gotten off into motor 5, all right? So they've gotten off just slightly rostral to it. So a lesion here, you wouldn't even interrupt the axons going to motor 5, but you don't have to worry about them anyway. So let's review. Middle cerebellar peduncle, you'd have an ipsilateral incoordination, ipsilateral. Pontine gray, contralateral motor incoordination. Cortical spinal, contra problems. Cortical bulbar, contra tongue, contra face below the eyes. Cortical spinals, contra hemiplegia. Move up into here, lateral meniscus, subtle auditory deficits. Move into here, ALS, pain and temperature from the face, or pain and temperature from the body. TTT, pain and temperature from the face, but not two-point. Medial meniscus, two-point, etc., from the contralateral side of the body. Descending autonomics, ipsilateral eye. Medial longitudinal fasciculus, we already know, we've talked a lot about that. It's got a long way to go, and it travels through many different sections, so there are lots of opportunities to interrupt these fibers. Again, they're headed for the ipsilateral medial rectus lower motor neurons. They're starting in the contralateral abducens nucleus. I'd like to say a couple words about the corneal reflex. When you touch your cornea, that information is going to come in over the fifth nerve. Let's say we touch the right cornea. It's going to come in like that. And we're going to send the light touch information into the chief sensory nucleus. Well, once it's in the chief, well, you know that if you touch one cornea, both eyelids close. So here comes the touch, comes into the chief, and you know that you then close both eyelids for you're going to be contracting those apicularis oculi. So how can we contract both eyelids? Well, information is going to come in about the touch, and then from here, it's going to go to both motor sevens. So let's do a couple lesions. And again, it's going to come in on one side, and then it's going to go to both motor sevens. The motor seven on the same side will help you with the direct corneal reflex. The motor seven on the other side will be the consensual or indirect. So if we had a lesion here of the fifth nerve, all right, we're going to get those sensory fibers coming in there from the cornea. If we had a lesion on this side and touch that cornea, you're not going to get a direct or a consensual. Again, if you have a lesion over here and touch this cornea, you're going to get both. If we go down to the motor 7 here, let's say we had a lesion in this motor 7 on the right. That's our only lesion. Touch this cornea, you're not going to have a direct. Touch this cornea, you are going to have an indirect. Touch this cornea, you're going to have a direct. Touch this cornea, you're not going to have an indirect. Okay, now don't forget, nothing coming in here is coming from the pharynx. Nothing coming in here is associated with cranial nerve 7, 9, or 10. So these are only trigeminal afferents coming in here, and your gag reflex is going to be fine. Also, if you go over here into the TTT and interrupt it, again, you're not going to have any 2-point in here, just going to have pain and temperature. You're also not going to affect the gag reflex. Now, if we move forward here, rostral to level 7, the identifying characteristic of this level is going to be the superior cerebellar peduncle right here. It's a big myelinated bundle. Here's the fourth ventricle. Here's the superior cerebellar peduncle. The other side, this is a cerebellum. So these axons are coming out of the cerebellum. We've talked about the inferior cerebellar peduncle. We've talked about the middle cerebellar peduncle and the pathways that are in those peduncles that are going into the cerebellum. Now we're talking about a pathway that conveys cerebellar information out, efferent, towards other parts of the brainstem and thalamus. So these fibers are starting in the cerebellum of the same hemisphere. So these would be the superior cerebellar fibers coming out of the left hemisphere. These would be the superior cerebellar fibers coming out of the right hemisphere. And they're going to go places rostrally in the brainstem, and we're going to watch them. But think about a lesion here in this superior cerebellar peduncle on the left. It's coming out of the left cerebellar hemisphere. You know that a lesion of the left cerebellar hemisphere gives you incoordination on the left side of the body. This peduncle is like having a lesion in the left hemisphere of the cerebellum. It's on the left, it's carrying information out of the left hemisphere. So a lesion here of this superior cerebellar peduncle would, like the cerebellum over it, give you an ipsilateral motor incoordination. So the peduncle is just like the overlying cerebellum when it comes to lesions. It's starting in the deep cerebellar nuclei. Those are just nuclei deep in the cerebellum that have cells in them, and the axons are running here as they go forward from the cerebellum. So a lesion here in this left superior cerebellar peduncle will be the same as a lesion in the overlying cerebellar hemisphere. That is, it's going to affect the ipsilateral side of the body when it comes to motor coordination. Now, we can watch this peduncle as it goes forward and crosses. So let's take that a little forward. Here's those same axons right here. Again, let's start here. They're going to go rostral, and they're in here like this, and you can see some of them are starting to cross. Okay, so this whole thing here would be coming from right here. So it's going rostrally, but they're starting to go a little more towards the midline and cross. So this would be the superior cerebellar peduncle. Now, if I was to ask you where these cell bodies are, you'd say ipsilateral cerebellum. If I was to ask you where these cell bodies are, you'd say ipsilateral cerebellum. If I was to ask you what the deficit would be if we interrupted it, you'd say ipsilateral motor incoordination. Now, some are crossing. I wouldn't ask you those, okay? So these definitely are still on the same side. Ipsy problem, and of course here the ipsy. So let's go. Here they are right here, starting to go towards the midline, and whammy. All of a sudden you get here to the level of the inferior colliculus, and you can see this big bundle of crossed axons is crossing in here. This is the decussation of the superior cerebellar peduncle. Now the two decussations, the two peduncles are crossing the midline, but you can see it much better here. So if I was to ask you here what would happen if you had a big lesion in this decussation of the superior cerebellar peduncle, you'd have to say incoordination on both sides of the body. Finally, you get a little rostral to the decussation. You can see all these fibers here and here. Now these are fibers that have actually started from the opposite side of the cerebellum. So let's go back. They've crossed here, they're crossing here. Now you're back where they started. So here we'd say ipsilateral incoordination. Here we'd say ipsilateral motor incoordination. Here we'd say bilateral motor incoordination. And here, now you have to think, these came from the opposite cerebellum. So you'd have to say, if I had lesions in here, you'd have to say contralateral motor incoordination. So let's go back a couple. So that's your superior cerebellar peduncle, and that's the identifying characteristic of this level. The rest of the stuff you should know, all right? So all these cells in here, pontine gray, pontine gray. Where are they going? Well, they're crossing in those pontocerebellars and going into the middle cerebellar peduncle. It doesn't look as big here. A lot of these fibers are going to have to go back a little bit to get into this middle cerebellar peduncle. So this is it. This is the most rostral part of it. Pontine gray still looks the same. Pontocerebellars look the same. Well, we got our cortical spinals in here, correct? If this is the right cortical spinal tract, you're going to have a left hemiplegia. We got our cortical bulbars in here, and they're going to go to where? They're going to go to motor 5. We don't have to worry about that. They're going to go to motor 7. So if you have a contralateral hemiplegia, you're also going to have a contralateral problem with your face below the eyes. Keep going down, we're interrupting those cortical bulbar fibers, the hypoglossal. So if you're going to have a contralateral hemiplegia, you're going to have a contralateral deviating tongue. You're also interrupting cortical bulbars here ambiguous, but you don't have to worry about them. There's a lot of stuff in here you already know, but remember, those cortical bulbars in here are destined to go down further. And we're getting all the important cortical bulbars. We're rostral to motor 5. We're rostral to motor 7. We're rostral to hypoglossal, and we're rostral to nucleus ambiguous. So what do you think's jammed in here? Well, all the favorite things that we learned down there, level 1, 2, and 3. So you're going to have a little bit of the lateral lemniscus out here. Subtle auditory deficits. But then we got the biggies in here. The ALS, well, contra pain and temperature problems. The descending autonomics, ipsy eye constriction. Medial lemniscus is in here, headed for the VPL, with thalamus, which is rostral to this, contralateral loss, 2-point, etc. And then you got your TTT in here. Now the TTT has the pain and temperature fibers, but don't forget, now we're rostral to the chief. We're rostral to the chief, so they filtered over in there and here. Once you're rostral to the chief, you not only think pain and temperature on the opposite side of the body, but you start thinking 2-point, etc. from the opposite side of the body. Then we got our old friend, the medial longitudinal fasciculus, better known as the MLF. It's headed for the oculomotor complex rostral to here, especially to those medial rectus neurons that are going to make the eye go this way. We also know that these axons are starting in the opposite abducens. So abducens have these small neurons which go up, project up here, and there they are right now, headed for the oculomotor complex, which is just a little bit rostral to where we are right now. Now here we have level 8. For me, the identifying characteristic is this decussation here of the 4th nerve, right here, which we'll talk about in a minute. We go down here, we can see accumulation of axons here on the ventral surface. We go back a level, we can see as we move from the rostral pons towards the midbrain, all these cortical spinals and cortical bulbars and cortical pontines kind of begin to aggregate here on the ventral surface. And in the next level, this will be even more distinct. But this we call the cerebral peduncle. Okay? A peduncle is just a bunch of axons. So a cerebellar peduncle would be something that's full of axons associated with the cerebellum. The reason that we call this the cerebral peduncle is that everything coming through here is coming from the cerebral cortex, corticospinals, corticobulbars, and corticopontines. And of course, this is going to have lots of corticobulbars in here. Those go into 5, 7, ambiguous, and 12. Lots of corticospinals, and of course, lots of corticopontine fibers, because most of the corticopontine fibers are going to get off back in here in this pontine gray. So let's talk about a lesion here in the right cerebral peduncle. It gives you a contralateral hemiplegia. Your tongue would go contra. Your face would be weak contra below the eyes. The uvula would be straight, and so would the jaw when you opened it, because both motor 5 and ambiguous get bilateral corticobulbar input. The upper part of motor 7 gets bilateral, but the bottom part gets contralateral, crossed. We have a little bit of pontine gray in here, right in here. We move up in here, all right, move up in here, we're going to have the TTT, all right. It's going to be carrying information about pain and temperature and 2-point discrimination from the face. We have the ALS, we're going to have the descending autonomics, and we're going to have the medial lemniscus all jammed in here. Now what's distinct is this little thing right here called the lateral lemniscus. The lateral lemniscus. Now this will be the one you forget. This is a part of the auditory system. It's ascending to the inferior colliculus, which is just rostral to this. This is where the lateral lemniscus really shows the best. Now, some of these cell bodies could be in the cochlear nuclei, could be in the superior olive. Let's go back and see. This is really distinct here. This is the most distinct place you'll ever see it. In here is kind of right in here somewhere. And here, right in here. Okay? All right, right in here. Now you know they're starting, it's coming out of these cochlear nuclei right here. So as we go forward, here's the lateral lemniscus. Here's the lateral lemniscus. Let's go back one. Some of these fibers in the lateral lemniscus, of course, could get out in the superior olive. Don't forget that. So here we go. Now it's right in here. Now watch this. Still right in here. And bang, right there. So I'd make sure you know this one because it's pretty distinctive. If I'm interested in asking the lateral lemniscus, I'm going to probably ask it to you right here because it really shows up well. Down in here are other friends that we already know. I think the hardest point here is that there's 2 points from the face in the TTT here. All the deficits here would be contra except for the constricted pupil. This, we don't have to worry about contra ipsi, we just say subtle auditory deficits. Move in here, we already know this is the superior cerebellar peduncle. Some of it's starting to cross. Okay, we go back, there it's starting. There it is right there. So here, a lesion here would give you a ipsilateral motor incoordination. All right, these are starting in the deep cerebellar nuclei on the same side. They're going to go forward a little bit more, cross, go into the rubra of the other side, and keep going up to VA and VL in the thalamus. But this would be your superior cerebellar peduncle, still uncrossed. Some of it's starting to cross down here. Here's your MLF. So these axons are heading up to oculomotor to those medial rectus neurons. You have a lesion here. You can't voluntarily turn this ipsilateral eye medially. In this particular case, if you had a lesion in the right MLF, you couldn't get this eye, this ipsilateral eye, to go to the left. Wouldn't have any atrophy. These axons are starting in the opposite abducens. So this little cell here is crossed, its axon goes up. Look at that, I don't have to move the pointer. Go right there. Okay, and we're going to watch this in the next couple sections too. You have a little nucleus sitting right here on the MLF. This is called the trochlear nucleus, cranial nerve 4. Trochlear. So there are cells in here, somatomotor cells, whose axons go up like this and cross. It's the only cranial nerve that exits the dorsal surface of the brainstem. Remember, all the other ones are coming down to the ventral surface. So here's the trochlear nucleus, and it's going to send its axons up and over, and then this rootlet's going to come down like this, join cranial nerve 3, and go out to the superior oblique, SO4, trochlear, SO4. Now, let's just say we're talking about this nerve right here as it's come out, and it's going to go to the right superior oblique. Now, this can do a number of different things for your eyeball, for the right eyeball. If you think about it, if you think about the anterior-posterior axis, if you think about what's going to happen with the, if you like stick a long fork from AP, from the front of your eye to the back of the eye, when you contract the SO4, it's going to cause the eyeball to intort, or medial rotation, intort, medial rotation. If you think about the vertical axis, a long fork going through the vertical axis of the eyeball, the SO4 is going to cause your eyeball to abduct, A-B-D-U-C-T, abduct. If you think about the lateral axis, a toothpick running from lateral to medial through the globe, it's going to, superior oblique is going to cause the eye to depress, go down. So let's say that we're looking at this right SO4 nerve. It's running out to the right superior oblique muscle, and it's got a lesion. So we've lost this right superior oblique nerve, and we have atrophy of the right superior oblique muscle. That right eye, because of the inferior oblique muscle that's still working, that right eye will be elevated and extorted, or laterally rotated. It will be elevated and extorted. Your left eye will be okay. It's probably looking straight ahead, so you're going to get double vision. Now, what you want to think about is how you can get the left eye to be looking at the same point that the right eye is looking. The right eye is elevated and extorted. So how can you get your left eye to elevate and intort by just the way you carry your head? Well, the way you could do this is you could tilt, not rotate, tilt your head to the left, which would cause, reflexively, your left eye to intort. Remember, your right eye is extorted. You want your left eye to intort so it matches up. So you tilt your head to the left, and then to get that left eye to go up reflexively, you kind of flex your chin. So if you had a lesion of the right superior oblique muscle, or the nerve, trochlear nerve, your right eye would be elevated and extorted. You would therefore then tilt your head to the left to get your left eye to intort, and you would flex your head down a little bit to get your left eye to elevate, and then you would get rid of the double vision. So what you would do is, if you lost the right, you're going to tilt and flex your head to the left if you lost the nerve. Okay? Remember now, because this is crossed, all right, if you lost this left nucleus, you would actually be tilting your head to the left, or ipsilateral. If you lose the right nerve, you're tilting your head to the opposite side, that is, to the left. Don't forget now, it's a lower motor neuron, so you're going to get atrophy. Now this is level 9, and here our identifying characteristic is right here, the decussation of the superior cerebellar peduncle. So if we had a lesion here, we'd have a bilateral loss of incoordination, bilateral incoordination. Don't forget where these axons came from. Here they are before they cross. Here they're starting in the superior cerebellar peduncle, coming out of the cerebellum on the same side. They then go forward. Some of them cross. Most of them are crossing right here at level 9. Now after they've crossed, some of these are going to go into the red nucleus. Some of them are going to continue to go up to VAVL, that is the motor thalamus. We look down here, we can see a great bundle here. Again, this is a cerebral, not cerebellar, cerebral peduncle. This is filled with cortical pontines because the pons is caudal to us. Corticospinals, you know, they have to go all the way down to the spinal cord. And all the cortical bulbars are in here. So if we had a lesion of this cerebral peduncle here on the right side, you'd have a left hemiplegia. Your tongue would go to the left when you stuck it out. Your uvula would be straight when you say ah. Your jaw would be straight when you open your jaw voluntarily. And your face would be weak on the left side below the eyes. So everything's left. A lesion here on the right cerebral peduncle gives you a left hemiplegia, left tongue deviation, left weakness of the facial muscles below the eyes. No problem with your uvula, no problem with your jaw, because motor 7 gets a very unique distribution. Don't forget the lower part of the nucleus gets a crossed input, the upper part of the nucleus gets bilateral. Remember, you can always wrinkle your forehead. That is with a cortical lesion. If you have a lesion in the motor 7, you cannot wrinkle your forehead. You can't wrinkle any of the muscles of facial expression. If we move up in here, we get to Substantia nigra, which is really something we're going to talk a lot about in the second module. Cells in here are involved in the production of dopamine. And these cells then project up to something called the striatum. So we call these nigrostriatal, and release the dopamine. Lesions in Substantia nigra result in Parkinson's disease. The classic characteristics of Parkinson's would be a resting tremor. That is a tremor when your hand is at rest. It just oscillates. Decrease in movement, slow movement, bradykinesia. And then your muscles are kind of rigid, and we call this rigidity. You also have some postural problems. Now, rigidity is very different than spasticity, and we'll talk about that later on in the course. Substantia nigra, Parkinson's, dopamine. These cells in here, when you cut the fresh brain, are black because of the melanin pigment. Very distinctive look. So cerebral, peduncle, Substantia nigra. Decussation of the superior cerebellar peduncle, no problem. What do you got in here? Well, you got our old friends, medial lemniscus, ALS, descending autonomics, TTT. And of course that old TTT's got pain and temperature, and it's got 2 point. And that, no lesion to TTT is going to give any problem with the gag reflex. All right. Now the ALS is headed for VPL. TTT is headed for VPM. The STT is going to be just up in here. All right. Now we get a lesion of the STT, that's going to give you an ipsilateral loss of taste. Okay. So the STT doesn't travel with this group of pathways that are very familiar to us. All these problems would be contra except for the eye, which constricted eye would be on the ipsilateral side. Of course, you wouldn't have any sweating in the head, but you also wouldn't have any sweating in the body on the ipsilateral side. This is something kind of neat. This is the lateral lemniscus going into what we call the inferior colliculus. Here's your big inferior colliculus here. Here's your other inferior colliculus. Here's the cerebral aqueduct right here, and something called the periaqueductal gray. But this is a typical midbrain section, okay? We're not in the Pons. This is the midbrain up here. And down at the bottom we have the cerebral peduncles right here. Then we have what we call the tegmentum, which would be the nigra, these pathways, decussation. And then we have something called the tectum, or the roof. Mainly the inferior colliculus right here, and at the next level the superior colliculus. So this would be the roof, or tectum. This would be the tegmentum in here, and this is the base of the midbrain. You'll see a little piece of the oculomotor nerve right here, coming out ventrally. Also see our good friends, the medial longitudinal fasciculi on each side. Again, these axons right here have started in the left abducens. They're going on up to the right oculomotor, especially to those medial rectus neurons. Here's a little piece of the trochlear nucleus. You can compare this, see it sits on the MLF. You can compare it with this one right here where you can see it. I don't know, I guess I like it better here. The reason I like it here is you can see the nerve going up and decussating. MLF, trochlear, inferior colliculus, lateral lemniscus going into, see this? Lateral lemniscus going into the inferior colliculus. So your lateral lemniscus, inferior colliculus, all you got to say is subtle auditory deficits. Okay? STT is kind of hard to see sometimes, but Those axons would have their cell bodies in the rostral nucleus solitarius. They're headed up to the VPM and they're carrying taste information. Well, here we are at level 10, so we're at the most rostral part of the brainstem, at the midbrain. Here you can see the tectum, or the roof. There's a superior colliculus up here. Here's a cerebral aqueduct, something called the periaqueductal gray right here. Here we can see a huge nuclear complex called the Oculomotor. Here's some of the MLF. Now, these fibers right here are part of the superior cerebellar peduncle after it's crossed, okay? So these axons are, some of them are going to get off into the red nucleus, which we call the ruber-duber. Some of these axons are going to get off in there, and some of them are just going to keep going forward up into the next part of the course, up into the thalamus, which is rostral, to the VAVL. Here you can see some of the rootlets of the third cranial nerve coming down like this. Here we have our old friend, the Substantia nigra, and our better friend called the Cerebral peduncle. We also have lots of old buddies right here, lots of old buddies. So in here, we're going to have the ALS, TTT, medial meniscus, descending autonomics, and then a new bundle right here. This is one that's going to cause you problems. It's called the arm or the brachium, arm brachium, same thing, of the inferior colliculus. Well, this is an auditory bundle. Remember the pathway that we learned back there with Kim a long time ago? Ventrocochlear up the lateral meniscus to the inferior colliculus to the medial geniculate body. Well, remember now, here the lateral meniscus is going into the inferior colliculus. So then a new pathway starts here in the inferior colliculus. These cells have axons that go rostrally, right here, is the arm of this, the arm of the inferior colliculus, or the brachium of the inferior colliculus. Now here's the superior colliculus, so the arm of the inferior is just kind of going down by the side here. And it's going to keep going until it reaches the thalamus, which is in the next level. That is the medial geniculate body and synapse. Then the medial geniculate body is going to send that auditory information up the cortex. So if we had a lesion here in the brachium of the inferior colliculus, you could say subtle auditory deficits. You had a lesion in the inferior colliculus, which is the source of that brachium, subtle auditory deficits. Lesion in the lateral meniscus, subtle auditory deficits. Again, our friends are in here, we know that. Nigra, Parkinson's, we're not going to be problem with that. Well, gee, what's in here? Corticospinals, corticobulbars, and corticopontines. If it's the right side, again, we're going to have left hemiplegia, left tongue deviation, a straight uvula, facial weakness on the left below the eyes, and when you open that jaw, it's going to be really straight, okay? Now we go up in here to the oculomotor complex right here. This has two divisions. One of it is preganglionic, parasympathetic, and the other is just plain old somatomotor. The somatomotor fibers, all right, are going to come out of here. They're going to have axons that come out, and they're going to go to every eye muscle except SO4 and LR6. So they're going to go to the levator, palpebrae. So, and to everything except SO4 and LR6. So if we interrupted those axons that are involved in the innervation of those eye muscles, first of all, the lid would be down because the levator, palpebrae, is shot. So you can't elevate the lid. So your lid is down. Now don't forget, you can also get a ptosis from interrupting these descending autonomics. But now we just can't get that levator up. So your eyelid's down, so you really wouldn't have any double vision. The only way you get double vision is if the two eyes are looking at different points in space. But here you only got one eye seeing anything because the levator is because the eyelid is down. If you were to raise the eyelid, you know that you got SO4 and LR6 working for you, so your eye is kind of down and out, but the eyelid would be down. Now you also have a preganglionic parasympathetic component coming out of here. Now those cell bodies are in something called the Edinger-Westphal nucleus, Edinger-Westphal, preganglionic parasympathetic. But those little cells have axons that are going to come out here with the somatic fibers, and they're going to go to ciliary ganglion, and from the ciliary ganglion they're going to go to the iris and going to cause, remember now this is parasympathetic, is going to cause constriction of the iris, of the pupil. So you can think if you had a third nerve lesion, sympathetics are going to take over, and that pupil is going to be really big, really big. So oculomotor complex has two parts, somatic and a preganglionic parasympathetic. Loses somatic, the lid's down, the eye is kind of out and down. Lose the preganglionic parasympathetics, the pupil is dominated now by the sympathetics, and the pupil is very large on the ipsilateral side. The other structure we have here is the red nucleus. We already know that some of the fibers in the superior cerebellar peduncle, now these axons here have actually started from the opposite deep cerebellar nuclei. So if we had a lesion in these axons here, some of these are going to go up to the VAVL on the same side. VAVL is going to go to motor cortex on the same side, and you know once you get in motor cortex, you're controlling the other side. So a lesion here in these superior cerebellar peduncle fibers will give you a contralateral motor incoordination. Now, the red nucleus is getting some input here from these fibers in the superior cerebellar peduncle, some input from the cerebellum. And these cells have axons which cross and go down through the brainstem in something called the rubrospinal tract. So we call this the red nucleus or the ruber. We also can call it the ruber or the duber or the sluber. And somebody actually gave me some beer called the Huber beer, so it's a ruber, sluber, duber, Huber. But these cells, again, have axons which cross, and we can't see where they're crossing, and we never see it in the brainstem. Can't identify this in the brainstem, but we call it rubrospinal. Once it gets in the spinal cord, it travels in the lateral funiculus. Now you have to know that. Now what other famous pathways travel in the lateral funiculus? The lateral cortical spinal tract and the dorsal spinal cerebellar tract. But these axons, these rubrospinal axons, travel in the lateral funiculus, and they travel just ventral to the lateral cortical spinal tract. And these axons kind of even terminate in the same parts of the spinal cord gray as the lateral cortical spinal tract. So investigators think that these rubrospinal fibers kind of work with the lateral cortical spinal fibers in controlling motor movements. But we don't know much about what happens with lesions in the ruber, because in humans it's not only going to get the red nucleus, the lesion, it's also going to get this superior cerebellar peduncle. Now you know if you get this superior cerebellar peduncle, you're going to have incoordination on the opposite side. Well, also think about a lesion here, the ruber. What side of the body is going to be affected? The opposite side. So it's very difficult to say, well, what's causing the motor problem? Is it the fibers of the superior cerebellar peduncle, or is it the ruber? So we just say motor incoordination problems on the opposite side, okay? Gets its input, this ruber gets its input from the superior cerebellar peduncle after it's crossed, and then there's some cortical fibers called cortico-rubral fibers that come in here too. I think the only thing left then would be the old superior colliculus here, superior colliculus. Not to be confused with the inferior colliculus. Well, how are you going to know this is the inferior colliculus? Well, you got the decussation of superior cerebellar peduncle. You can see the lateral lemniscus coming into it. All right. This looks a little bit different. In a Nissl stain, where we're looking at cell bodies, this would have many different layers in it. Also, the superior colliculus is more rostral in the midbrain. So you can see the ruber here, the oculomotor, and the superior colliculus. If we go back here, you can just see the decussation, even some trochlear and inferior colliculus. So that's one way of telling them apart. The superior colliculus is a structure that gets input, visual input from the retina, and but it also gets some other inputs like auditory inputs, I'll say from the brachium here, or even some somatosensory inputs. But most people always usually consider this to be a visual reflex center. And how this works is that visual information from over on this side. Now you don't know the organization of the fibers in the retina, but let's just say that a visual stimulus falls over here on the left and in the left side of your visual world. Well, it's eventually via the retina going to come and turn on cells here in the right side, in the right superior colliculus. So here's your visual stimulus. It eventually will reach here over pathways that you don't have to worry about. So a stimulus over here is going to turn on this superior colliculus, which is pretty much a reflex center for eye movements. So visual stimulus over here turns this colliculus on. Well, one thing is you probably, if there's a visual stimulus over here, you want to reflexively turn your head over this way, and your eyes, but let's just think about turning your head that way. Well, here's the visual stimulus, turns on this superior colliculus, and there are fibers that come down, and we can't see them, and descend down through the brainstem in something called the tecto, T-E-C-T-O, tectum meaning the roof, tectospinal tract, and it's going to go all the way down the brainstem into the spinal cord, and it's going to travel in the ventral funiculus. Now don't forget, you know some people in there. You know some pathways in the ventral funiculus. But the tectospinal pathways in the ventral funiculus are especially going to go to the cervical regions and going to turn on neck muscles that enable you to turn your head towards the visual stimulus. Now these neck muscles wouldn't include the sternocleidomastoid. Okay, don't think about that. Just think about neck muscles that would turn the head towards the visual stimulus. So visual stimulus here turns on the superior colliculus, turns on the tectospinal tract, goes all the way down to the cervical cord, turns on lower motor neurons that enable you to turn your head towards the visual stimulus. So finally, we just have this structure right here, which I know everybody's just been dying to talk about. This is the periaqueductal gray. Periaqueduct, periaqueductal gray. There are cells in here that project down the brainstem, but first let's talk about what happened in some experiments in rats where they stuck some electrodes in here and they started stimulating the periaqueductal gray like crazy. They found that these rats, you could actually put them on a hot pad and they wouldn't even want to get off because they were feeling so good, because this periaqueductal gray sends projections down to the brainstem to a nucleus called nucleus raphe magnus. So let's say there's a cell here. It projects down to level 3 to something called nucleus raphe magnus. Now, it turns on nucleus raphe magnus, and nucleus raphe magnus in turn has a projection down to the dorsal horn. And that this nucleus raphe magnus, these cells are serotonergic. So, all of a sudden you turn on this cell, it goes down to raphe magnus, turns on raphe magnus, which is serotonergic, and raphe magnus is going to have an axon that goes down into the dorsal horn. And it's actually going to inhibit those pain fibers as they're coming in to the dorsal horn from those C's and D's. So by stimulating or turning on these cells, you turn on raphe magnus. Raphe magnus then goes down and inhibits the transfer of information in from those C and D fibers. So it kind of blocks out the pain. Well, you might say, well, what turns this on, you know? Well, we don't know, but we do know that our emotional brain, the limbic system, has pathways to the periaqueductal gray. We also know that these periaqueductal gray cells, these periaqueductal gray cells, have receptors for some of the endorphins. So the pathways into here are going to be using endorphins to turn on these periaqueductal gray cells, which have receptors for endorphins. Once you turn this on, you go down, you turn on raphe magnus, serotonergic, turns on those axons which are inhibitory on the transfer of the information from the C and D fibers into the dorsal horn. Now I want to go back through here and just do a little bit of reviewing of some of the structures I think are the hardest. I think out in here, this is STT. You have to be careful. Sometimes I know I get a little sloppy pointing it out. So it's probably in this area right here. Cell bodies would be in the solitary nucleus rostral part, and it's headed up for VPM, solitary ortholamic tract. You'd lose taste on the ipsilateral side. You know everything out in here. go back a little further here, we're going to have the STT right about in here. I also want to go up in here and talk a little bit about a lesion of this right medial longitudinal fasciculus. If we have a lesion of this right MLF, we cannot get the right eye to go medially when we try to look to the left. So this is the right eye right here. We cannot get the right eye to go medially or to the left. Now, I mentioned that with lesions of the MLF, you can get nystagmus. So let's do this very slowly. Put your two hands in front of you and extend the palms, and so you got a right eye and a left eye. And think about the globe in the right eye. When you want to look left, that right eye won't go left. It won't go medially. So just kind of do your hands, and your left globe is going laterally pretty easy to the left, but the right eye won't be able to go across the midline. Now, what the brain does is try to figure out a way to get the right eye to go to the left, and it uses the convergence center. Now, convergence is just when both eyes go medially at the same time. So do that. Just say, well, I'm going to have both of my hands go medially at the same time. All right, so that's convergence. So let's say with a lesion of the right medial longitudinal fasciculus, that right eye is trying to go medial. The left eye can go lateral by itself. So what happens is that convergence center is going to try to turn that right eye medially because you can't do it through the MLF. But the convergence center can only affect both eyes at the same time. So when the convergence center kicks in, it's going to turn your right eye medially, but it's also going to turn your left eye medially. And you're trying to look to the left. So what happens is the convergence center kicks in, turns your right eye medially, but don't forget, you can turn your left eye laterally. Okay? So you look left, that left eye goes flying out, the convergence center kicks in and moves your right eye medially, but then it's trying to also move your left eye medially and brings it left eye back slowly. But then the voluntary action pops it far left. So what you have is that left eye jumps out laterally because it's innervated, but then that convergence center pulls it back medially, pops back left, slow to the right, pops back left. So there's your nystagmus. So it's slowly pulled medially by the convergence center, pops back left by the voluntary control. So the fast movement, so it's the good eye that's got the nystagmus. The slow movement is medial, done by the convergence center. Fast movement is lateral to the left, done by voluntary control. So the good eye has a nystagmus, and the direction is in the direction of the good eye. So we've talked about the MLF, the trochlear. I think this trochlear one is probably one of the hardest. Don't forget you have to figure out what muscle is bad. Now here we're in the nucleus. So it's going to go like this. So this muscle is going to be bad. The left muscle. And then you realize that you have to tilt the opposite way to the muscle. But if you were up here in the nucleus, you would be tilting ipsilateral to the nucleus. On this one, I think this is going to be the hard one. The lateral lemniscus. All right? This is going to be the hard one. It's headed for the inferior colliculus, subtle auditory deficits. On this one, it'll definitely be the superior cerebellar peduncle. I'm not going to ask you the lateral lemniscus. I want you to know the superior cerebellar peduncle. That's the one you'll miss. On this one, you'll miss the chief. This is the chief, this is the motor. Remember, motor is medial, M and M's. Motor, M. Medial, M. You'll miss this. You'll call this the motor or something like that. You might also think that there's some gag reflex fibers in this. On this one, you'll say this is motor 7 instead of the superior olive. You'll say this is superior olive instead of motor 7. Facial colliculus, think about what would happen if you had a lesion of the facial colliculus. Well, you're going to get these fibers coming up from motor 7, so you're going to have a paralysis on the ipsilateral side of muscles of facial expression. You're also going to have a problem here with abducens. And if this is the right side, and you have a lesion here, you're not going to be able to turn your eyes to the right upon voluntary control. Okay? So don't forget, you're going to have some motor 7 problems and some hyperacusis. You're also going to have some eye problems. On this one, oh, I don't know what you'll miss. You'll probably wonder what this is. And of course, you know that the medial vestibular nucleus gives rise to the medial vestibulospinal tract. Remember, most of it's ipsy, some of it's crossed. You're going to call this the MLF, which it is. Now, what's confusing is this is called the descending medial longitudinal fasciculus or the medial vestibulospinal tract. This is the ascending MLF because it comes out of abducens. Don't confuse it with this right here. This is actually, this pathway is starting here. So we call this a descending MLF. Or we should just rename it just the medial vestibulospinal tract. On this one, oh, it's always solitarius. People always forget this little solitary tract. And of course, accessory cuneate. That one is notorious over the years. On this one, I don't think anybody will miss anything. Maybe internal arcuate fibers. I don't know. On this one, oh, since we've gone over this so much, what would people miss? Probably, sometimes they get this confused, the spinal tract 5 with the dorsal spinal cerebellar. And really, this is the best place to see the two, dorsal spinal cerebellar here, spinal tract there. So I guess that does our review, and I hope everybody does really well on the exam.